Translation is the production of a polypeptide using an RNA template. Now, this RNA template that we're talking about is something that comes from the transcription process. So if we were to just write an example of an RNA template here, notice that whenever we have an RNA template, we're going to have a series of RNA nucleotides. They tend to be written in groups of 3 because recall that these groups of 3 are the language of the cell, or in other words, we refer to these as codons. So if we look at this in more detail, each one of these is a codon, and what those codons do is that they specify an amino acid. So each codon is going to tell the cell what amino acid should be placed in the polypeptide that we're trying to make. And that's what the translation process is translating. It's basically interpreting this language that is inside the RNA molecule to make that polypeptide product, which is going to go out and function inside of the cell. So if we look at how we're going to use this genetic code table, let's take a look at the table now and use this RNA sequence. So here we have the genetic code table, and this table is going to tell us what every single one of those codons means inside of the RNA molecule. So this is my RNA molecule, and if we go and we were to look up the very first codon that we have here, this codon codes for, as you can see here, AUG codes for methionine. So what that is telling the cell is that during the translation process, that is going to be the very first amino acid that gets attached inside this polypeptide. Then the cell would look at the very next codon, which in this case reads CCC, and so it would put in whatever amino acid was appropriate for that, and that's going to be proline. Now, as we start to hook together these amino acids as we go through here, keep in mind that these links that we're making, those are the peptide bonds, because peptide bonds are the bonds that we find between amino acids, and peptide bonds also are formed by dehydration reactions. So if we're forming a bond by a dehydration reaction, there is going to be a water molecule that is removed during that overall process. So every time we hook together a new or hook on a new amino acid, there is going to be a water molecule that gets released. If we look at the very next codon, GAU, GAU stands for aspartic acid down here, ASP. And again, another peptide bond, and then we would look up the last one, CUU, and CUU is going to code for leucine. Now, typically we would have a stop codon at the very end of a polypeptide that we're making. This one I'm just drawing very short just for illustration purposes. So in this case, we don't actually have a stop codon there. So what we have made right here, this is the polypeptide. That will be the overall end product of the translation process. If we take a look, notice that we read the 5' end of the RNA molecule first. So we're going to read the RNA 5' to 3'. And we also have a unique polarity to our polypeptide as well. On the very first end that we synthesize, this is going to be the amino end, or the end that has the amino group on it. Sometimes it's called the amino terminus. And then over here on the leucine side, or the last part that we make, what actually lines up with the 3' end, that's going to be the carboxyl end, or the carboxy terminus. Okay, so I'm just going to write that in there. And so if we use the same kind of terminology down here, we read the RNA 5' to 3', and we make the polypeptide amino to carboxy. If we talk about some of the molecules that are going to be involved in this overall translation process, the ribosome is typically the first one that comes to mind for a lot of people. And the ribosome is composed of 2 subunits, and each of those subunits is made up of some RNA, which we tend to call rRNA, and it is also going to have a protein component. And it is the RNA that tends to have the enzymatic or catalytic activity. And so this particular RNA that makes up the ribosome, we do tend to think of it as a ribozyme. And then the protein component obviously is very important for the overall structure of those subunits. So the ribosome, this is the kind of the coordinating component. It's going to coordinate all of the activities. If we were to draw the overall structure of the ribosome itself is made up of a large subunit, and then we also have a small subunit here as well. So if we draw our small subunit on here, this is going to be my small subunit, and the two of them, they will come together when they do actually function inside of the cell. There are also going to be 3 active sites inside here, and we call them A, P, and E. And each one of those is going to have a specific role in the overall translation process. And right between the subunits, right down the middle here, that will be where we actually have our RNA molecule or RNA template running through it. So let's take a look at some of the other parts of the overall translation process that are going to be participating. tRNA molecules are going to be special types of RNA molecules, which are called transfer RNAs. And the transfer RNAs are responsible for actually interpreting what the codons mean. And so if we look at a tRNA, the tRNA here I'm showing in green, and the tRNA itself has kind of 2 ends to it. On one end, it has 3 nucleotides in a row, and these are referred to as the anticodon. The anticodon is going to be complementary to one specific codon. And then on the other end, up here shown in purple, there is a place for an amino acid to be attached. So I'm showing here the purple is representing the amino acid, and this will be one specific amino acid that gets attached. So we will have lots of different tRNAs actually inside of the cell, and these tRNAs each have different anticodons on the end because these match one specific codon. And if you recall, we have 61 different codons that actually code for amino acids. So you would have to have a different tRNA to actually match up to each one of those codons that we have. So the tRNAs, depending on what the anticodon says, it will have a specific amino acid attached. There are enzymes inside of the cell whose job is to actually attach the amino acid. These enzymes are called aminoacyl, tRNA synthetase. They're very specific enzymes, but their overall job is to load the amino acid onto the appropriate tRNA. So what will happen during the overall translation process is that the ribosome will be coordinating everything and reading the RNA template, but the tRNA will be the one that actually looks at the codon and contributes or offers the proper amino acid to the ribosome so that the ribosome will be able to actually hook those amino acids together. And as soon as the amino acid gets used for translation, then an aminoacyl tRNA synthetase will need to reattach another amino acid to that particular tRNA.